Hello, and welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the first episode of Plantopia for 2025, and I'm excited to welcome Dr. Kranthi Mandadi, professor of plant pathology and microbiology at Texas A&M University, who is stationed at the Texas A&M AgriLife Research and Extension Center in Weslaco in South Texas. Dr. Mandadi completed a BS at the ANGR Agricultural University in India, an MS in Plant and Soil Sciences from Texas A&M University, Kingsville, and a PhD in Molecular and Environmental Plant Sciences from Texas A&M in College Station. Dr. Mandadi completed his postdoctoral training in plant virology also at Texas A&M University, before starting his position as an assistant professor in 2015. Dr. Mandadi's research focuses on fastidious plant pathogens, including the causal agents of citrus greening disease and zebra chip of potato. His work, which includes over 70 peer-reviewed publications, is highly cited and has been covered widely in the popular press. Dr. Mandadi has generated more than $8 million in research funds and over 25 combined patents and invention disclosures, all while training more than 36 undergraduates, 24 postdocs and research associates, and more than half a dozen graduate students. Dr. Mandadi is an active member of APS, serving as associate editor of phytopathology, and an active member of the APS Caribbean Division. He is the 2024 recipient of the APS Syngenta Award, which was awarded on his breakthrough basic and translational research of fastidious plant pathogens. Kranthi, thanks for joining us today.
Well, thank you. Appreciate the invite.
You're a full professor of plant pathology and microbiology at TAMU, Weslaco, where you and your research group are working to understand fastidious plant pathogen interactions and develop biotechnology in and around that topic. Before we get into all that good stuff, I want to talk briefly about Texas itself. It's a massive state. It's second in size only to Alaska. It's got 31 million people or thereabouts. The world's 8th largest economy, the most farms and highest farm acreage in the United States. Granted, mostly beef cattle and cotton, greenhouse and nursery crops, but still the second highest agricultural revenues behind California. What attracted you to Texas in the first place?
So my entry into Texas was just pure coincidence. I was born and raised in southern part of India. I did my bachelor's degree in agricultural sciences there. So I didn't know about Texas until I came to Texas. Once I came to Texas, I came to know how big it is, how diverse in terms of the ag ecosystem is. As you just said, there's a lot of rural farming communities here. Where I am in South Texas, specifically close to the border to Mexico, we are a very subtropical and tropical climate. Pretty much the same weather that I grew up back in southern India. So we have a lot of crops. So it was very good for me, to be in, Texas in all these years. I consider myself a Texan here now. My career since moving to the United States was within the A&M system. It's just big and there's so many opportunities. So I ended up staying here.
Right. Yeah. And I saw that, doing a master's first, then a PhD, postdoc, and now where you're at is a full professor, having worked up those ranks. Now, Texas A&M has a main campus in College Station, a branch campus in Galveston, a branch campus in other places around the globe, other places around the U.S., including Washington, D.C., professional schools and campuses in Fort Worth, McAllen, health campuses in Corpus Christi, Dallas, Houston, Round Rock, Temple. You did your master's in Kingsville, correct?
Right.
So there's clearly a campus there.
There is, and we call them the sister campuses. So as you just said, all the campuses are there throughout the state of Texas, mainly to care of the different geographic regions of Texas, both, in terms of the environment, there is differences, all the way from I think in the USDA hardiness zones, we are from 6 to 10, all the way, very cold to, to tropical climate. So yeah, Kingsville campus is a sister university campus. I got my master's in plant and soil sciences. That was my area of specialization.
And that's close to Corpus Christi, where that campus was, right? And that's
correct.
like on the Gulf Coast.
Yep.
Okay,
So 1.5 hour drive, I think, pretty much to the coast.
right. Now you are a full professor of plant pathology, and you are stationed at a Texas A&M AgriLife Extension Service District 12 headquarters. That's correct?
Yeah, so basically it used to be the state experiment station. So they renamed them as Texas A&M AgriLife, several years ago. So we are a part of the state agency, affiliated to the university system. So my academic appointment is in the Plant Pathology and Microbiology department, which is on the main campus, which is in College Station. And the state experiment stations, they are spread out. I think we have 13 centers throughout the state. South Texas, specifically in Weslaco, is a station here, both research and extension. So I'm 100% research appointment, but we do a lot of research as well as extension activities and collaborations as well as on our own, even though we're 100% research, we do
Right.
a lot of interactions with the growers, service, mostly applied translational research.
But your position itself, even though you're at an extension facility, you're not an extension faculty member, even though you do a lot of extension.
That's correct.
Clearly, the location might play a role there, but I also look at like the technical aspects of the kinds of questions you're asking and answering. And I wonder if in your lab, postdocs make more sense because of how technical and at the cutting edge your research actually is. And it requires a good foundation of that kind of technical knowledge.
Yes and no. Yes, because so see, we are being off campus to graduate students to come enter into the labs. It would take at least a year and a half, and it has come down to a year now. By the time they finish their coursework in College Station in the department, and then move to my lab, previous graduate students took up to 2 years sometimes. But now that kind of sets slow in terms of research project picking up on things in the lab. So which is why, most of the research faculty or even extension faculty sometimes have postdocs as a primary research body. Of course, we also have research assistant and associates as well. Now, having said that, graduate students are really, I feel like, they're the among the most motivated also in terms of idea creations and generation. So I would love to have more, it's just that it's just far from main campus. And that's why I just by necessity, I take more postdocs.
hmm Yeah, we have a similar situation where our fruit research station was in the eastern part of the state, and we had faculty assigned out there, but we're having a hard time recruiting students because they had to be away from their cohort, and also like developing that community is a big part of the grad school experience.
Absolutely.
So one of the other things that really caught my eye, and I'd like to talk to you about because I don't get many people on here that are so prolific in this regard, is that you have a lot of patents and a lot of intellectual property, invention disclosures, some 25, 14 patents and 11 invention disclosures. What's the difference between those?
So the invention disclosures are like the first step when you just have a, potential intellectual property that could have a market value. You would have to disclose that to the university. That is invention disclosure. That's a university level forms that we need to fill out. But patent applications and patents come later, depending on, if the university decides to go with filing a patent application, and then you have to prosecute and get up to the patent stage, which takes 4 or 5 years sometimes. So that's the difference. Now, if you ask why I have so many, I'll tell you the research that we are doing here in terms of being off campus. I do balance a lot of some of my basic research interests in terms of plant genetics and plant pathology and microbe interactions with translational research, which means, the findings that I generate from my research, how can I use it to benefit the growers? And the only way at least we as scientists can take a research finding to a product, to all the way to product, is through in crop protection world, it's mostly through technologies, and that could be potentially registered, pest control products or disease control products, right? And so
Right.
for that, we require a lot of industry, support and engagement. And And then if it's a disease control product, if I had to invite an industry to pick up, the research findings and take it to commercialization, they need to, of course, evaluate their market return on investment. For all of that to happen, you need to have an IP protection. A company would not pick up just for the sake of science, in terms of technology commercialization. So I was really fortunate when I started here about 10 years ago, my academic mentorship committee had senior professors, Dr. Eric Mirko, who unfortunately passed away around 2018, and he's my closest collaborator as well. So I learned a lot of these, how to protect IP, and that you can use later to advance your knowledge, or in this case, the discoveries to the field products. So I learned from him, and with him, I really, the first few years I was here, we discovered some new peptides. We call it antimicrobial peptides.
Right.
That could be, used as a broad spectrum for efficacy. They have efficacy against multiple bacterial or fungal pathogens. So, but how would you use that to take it as a product to control diseases? Is I learned from him, and then that you have to protect it first. Papers, yes, later, but first protect it, and then you would then have to, basically do all the experimental research and go with showing that these really work, and then, invite companies to take upon to the next steps of commercialization. So I was fortunate to have that experience, and then I've been continuing since then. I've been mentoring my students and postdocs in that process of thinking. Oftentimes we're, pretty much focused on the science part of it, and as a scientist, we're trained for that. We end up losing the sight of what is next after publications. What's next? Is something we should think of, right?
Right. Well, one of the things I think about is that I have one patent. And it's not a big deal or anything, and nothing ever came of it. But I know that when I was at Virginia Tech and worked through their intellectual property office, I know that there's the time barrier, which it could take a while, but also there's like the legal paperwork and stuff. It's like the numbers probably changed, but I remember at the time they're like, oh, it's $10,000 to file this patent. And of course, if you're filing a lot of patents, that's a huge investment with not a full understanding of the potential return on investment. So my question, I guess, for you is, if a company sees interest in your invention disclosure, do they often cover the costs of the filing of the patent with exclusive or at least first right of refusal to those technologies?
Yes. So the universities usually have technology commercialization offices. So at A&M, we do have one for the whole system. They cover all universities as well as state research centers. So they manage all our intellectual property. So yourself, you could go and look for potential companies that might be interested in your technology, and then approach them. But with making sure that you don't disclose things before you protect, don't talk to anyone. That's one thing that I was told, that once it's a public disclosure, without protection, it voids some of the prosecution of the patent itself. And once the licensing office or the technology commercialization office starts, looking into actually talking to the companies, they can come up with some terms, in which they can say, okay, you would pay some licensing fees. Here is the cost, and you would, also cover all these patent expenses. And sometimes there are multiple, not only in US, they have to file in other countries as well. So worldwide.
Right.
And so they have to pick up prosecution of all of those. They have to pick up sometimes attorney fees, depending on the motivation of the company to invest in your product. You could have that negotiation with your licensing office. And down the road, once the patent is filed, you have to also maintain it for a period of, I think US it's 25 or 30 years, right? And so every year there is an annual fee. I think they also, have to cover that for as long as they have interest in licensing the technology itself. So yes, companies can support it. Sometimes universities also pick up on their own, like
Right.
the technology commercialization office, they can do on their own, which depending on the framework of that office, if they see a real potential in that technology to become commercialized, they would rather invest now than to find a company, and wait for it, or lose the IP to somebody else, down the road.
Right. Okay. Yeah, thanks for that. I think it's an important discussion to have because, like so many things in academia and within the university framework, but also this applies to industry too, is and maybe those in industry get more of a lesson on this, but as academics, we're not trained for a lot of these things. We don't know. I mean, yes, that's why we have intellectual property offices. But like, if you never filed a patent or thought about it, you don't even know who to talk to or how to talk to them about. You don't even know if you have an idea that can be patented.
Right. No, yeah, absolutely. I think that's where mentoring, could help, where if you have somebody in your university, in your department, somebody already doing it, maybe time to learn from them.
Yeah.
And maybe at the graduate school level also, we need to kind of have a overview of this process to the graduate students so they have a better idea before they're going into postdocs or any other labs. So I think yeah, as a academic, just labs alone, we're not well trained for that. Once you move into the industries, yes, industries absolutely are really good at this. And in fact, some of our licensees, industry members that have licensed our technologies, they often tell us that, okay, before you just go and publish, think it through, just, make sure that if there is a potential for commercialization, show it to your offices, licensing offices, so they can also, help us out whenever, if you have any questions, you have any industry friends, talk to them.
So just to play the devil's advocate here, there may be like some that say, well, a patent pursuit seems to be something that would slow down the progress of research, not expedite. But, I think if someone took a clear view on these things, they could see that industry funds could expedite it because you're throwing more money at an issue. What's your view on that? Like view that pursuing a patent is going to slow down the pursuit of the answer to some of the pressing questions in our field.
I think what you said is true. It does slow down things in terms of actual dissemination of your findings, in terms of discoveries that you have done in the lab. These days we know how important it is to publish in prepress, preprint, archives, and just get the knowledge out, right? It is a challenge, especially also graduate students, if they're involved, they have a timeline. They need to walk through some publications, or even you're a faculty, junior faculty member that you need to go up for tenure. You need papers to is in showcasing in your package, right? It is a challenge, but in this case, the way I see it is, it's about, risk and reward. So the risk is, yes, it will slow down on getting this, paper out, but the reward would be much greater if it's protected. And then there is a company that you already know that would like to invest in your research. That brings you back, your research dollars back into it. And as well as the most important part is, that's the only way to get the product to the market. So I balance it with different projects we have in the lab. And I just shared earlier, some are towards the fundamental science, just exploration in terms of genomics or genetics of plant microbe interaction. So those, I don't anticipate much, technology products, but more of a knowledge base. So I built my program such that I have an influx of good papers coming out, as well as these high risk, high reward projects where I have some knowledge that could be potentially becoming a product. So
Yeah.
those take a long time.
And that's clear in looking at like your outputs. I mean, you've published a considerable amount, particularly when you consider that you went from assistant professor to full professor in 9 years or something like that, which is, a very expedited timeline. But I can see that you are balancing a nice publication and patent. So that's great because it's providing diverse opportunities for your trainees.
Absolutely. Thank you.
Now, you work on citrus greening disease, also known as HLB. I won't butcher it and try to pronounce the name of the disease, but this is an insect vectored gram-negative bacteria. It's in the same bacteria family as Agrobacterium, the causal agent of crown gall, and some rhizobia. The known vectors of HLB, the Asian citrus psyllid, the African citrus psyllid, psyllids themselves present some challenges. So I'm going to go ahead and assume that South Texas is a good area for growing citrus.
Absolutely. Specifically where Weslaco is, we call this the Rio Grande Valley. This is the only place we grow citrus commercially in Texas. So, and which is why, we actually had to work with some of the needs of our stakeholders, our growers. Citrus greening, as you said, it's a bacterial disease, and it's here, and it's also mainly first discovered, at least in US, in Florida, Since then, it has spread to most of the citrus producing states, including California and also Georgia, I believe, and it's expanding. So yes, we grow it here.
Right. And Texas is pretty high up there. What are they, third in citrus production in the US?
Actually, not that high. We are right now number three. California has took over Florida. We grow about, I guess, 20% of our production and our U.S. production is from Texas, but it is a significant amount, and it's almost, I think, at least 500,000 acres of citrus is at risk with this disease. So it's really important for our growers. I mean, and there is other problems in here, as we just talked earlier. The freeze, sometimes we get these deep freezes that takes us by surprise. 2021 we had a big freeze and that took down our citrus industry quite a bit. We have water problem. There's other problems, but it will be still on the mind of the growers.
And the other thing that I think it's good for the listeners to understand is that even though you produce less citrus than California and Florida, and I know Florida has declined in recent decades because of disease pressures and other factors, the Florida citrus market was largely for juice, whereas California and Texas are like whole fruit for shipping across the nation for grapefruits and other citrus. So maintaining the integrity of the fruit is really important because it's for fresh market. It's not for juicing or things like that.
Absolutely, absolutely. You said it right. So the disease or even any other stresses, the impact on the fruit quality hurts more the fresh fruit market than juice market. So yes, we are very, anything that impacts the fruit quality and yield for our growers is a problem here.
Now, the bacterial pathogens that are vectored by these true bugs are fastidious. And basically, for the listener, that means that they cannot be cultured outside of an insect host. However, I noticed that you have been developing, in your group, have been developing some techniques, some innovative systems for culturing these. Do you want to tell us a little bit about some of the progress your group has made in that regard?
Yeah, absolutely. So yes, what you described, the pathogen class is fastidious or unculturable, or we can say they are obligate host-dependent organisms that cannot survive outside their host environment. So yeah, when I first started in 10 years ago in Weslaco, when we came to know about this problem, and greening is caused by a bacterium that is an unculturable pathogen, that itself put me on a brainstorming session in my brain. So that how can we even work with a pathogen that's unculturable? I haven't worked with them before. In my postdoc and also in Texas A&M with plant virology, I studied about viruses being unculturable, but we can still get virions on some level, infectious clones out of it and study at the reverse genetic level. But this is the first time, and I started here as a faculty working with unculturable bacteria. So we talked with my colleagues here that have been already working with citrus greening, and I asked them, a question about, so then if it's unculturable, how do we study, effects of, therapy or a gene on it? So the way it's done currently is using, whole plant experiments, which means
Right.
if it's a gene testing, you would have to generate a transgenic plant. Engineering a citrus would take at least a year or a year and a half to get a citrus transgenic plant. And then to challenge it, you have to use insect vectors and so on to expose the plants carrying the bacterium and wait 6 months or even a year to test one genetic construct. You would take at least a couple of years. More or less same goes with testing with any therapies that you have potentially could be used against greening, like small molecules or chemistries. You would have to grow greenhouse plants and spray the plants or do delivery of that molecule to the trees and then look for alleviation of the symptoms, whatever the other metrics you are looking at. All of that takes 1 to 2 years still in the citrus gene testing or therapy testing.
Yeah.
When I started as a junior faculty, and then that means if we had to screen, let's say 100 molecules or 100 something, you would spend pretty much 5 years to 10 years. And still there would be, unlikely that, would complete all the experiments. So we really brainstormed it. And our short story is that we came up with this hairy root based co-culturing system where we're not culturing the bacteria out of the plant in test tube, but we are able to grow it in these hairy roots. Okay, hairy roots are basically, in plant biology terms, they are adventitious roots that we can induce on demand from any aerial portions of the plants. In this case, if it's a citrus leaf that is infected with this bacteria, you could induce roots or generate roots from that leaf. And the nice thing about it is that roots are advantageously, when you generate, you could add gene constructs to it through a binary vector, the same way you would do a plant transformation. So delivery of your genes and or even small molecules can be done very easily in these hairy roots that end up hosting the bacteria. The bacteria is going into these roots just because they are sink tissue, and the bacteria itself is in the sink organs. It's present in the phloem tissues. Phloem is connected to the roots and the leaves that we use to induce roots. So we created an environment there in the form of hairy roots. We call it a biological vessel for essentially asking the bacteria to use that to grow in there. So we can add all our testable therapies to these roots, and much faster and in a high throughput manner. We could scale this. For example, we could do 50 genes, for example, in a span of 3 months.
Oh, wow.
In a small molecules, we could do in a 48 well Petri dish, we could screen about 48 chemistries in 3 days.
That's amazing. Yeah.
And so that's the advantage of the hairy roots as a way to speed track some of these culturing bottlenecks for this bacterium. Now, it is not an axonic cultures. It is never an axonic culture. It is still growing in the host environment. Our goal was to move beyond that bottleneck and figure out, what's next, how to use it to screen and find the therapy. So to that purpose, it serves great, and we have used it. We identified many candidates, genes, peptides, and small molecules that you can see in some of our publications, as well as patents already, that are making headway into even the registration processes.
Well, thank you for that. Now, in reading about HLB, I see that there's Liberobacter asiaticus as well as the Africanus, and they have different temperature optimums. Which one is actually here, or both here, or just one?
Asiaticus is the one that's in the United States, and for the most part, there are many, many strains of Asiaticus. I think there is at least the ones that are in Texas, and there are some in Florida. And the Africanus is more there is also Europeanus, I believe, and then there is also another related, but the host range is also different. It's Solanacearum, Candidatus Liberibacter solanacearum. And we have it also in the US here, but it doesn't infect citrus. It infects all the Solanaceae family. It is transmitted by a different vector, insect vector. It's not Asian citrus psyllid, but it's
The potato psyllid, right?
right. There you go. So,
And that causes zebra chip disease.
yep, there you go.
Yeah. But one question I have is, I have to admit, I don't know much about these things, and I read up a little bit. I read that Africanus is asymptomatic at temperatures above a certain threshold. So if you're growing citrus in that temperature optimum or that temperature range, wouldn't it just go undetected because it's asymptomatic?
Yes, it's a big problem, in fact, for detection, early detection, for example. So a lot of times in addition to the seasonal fluctuations, the bacteria, it's still very, to me, finicky to grow in the trees. It doesn't accumulate that high levels. It's very, slow to reproduce, multiply. And we did some metabolic modeling and modeling analysis with the collaborators in Riverside and other universities. They, I think, predicted I guess duplication time could be anywhere from 60 hours to 80 hours. Still, it is a challenge that, because of its poor growth in the host tissues, even in its own native host citrus trees, detection is a big problem. And that has ramifications, because once a growers start new planting, for example, and they won't be able to even determine whether it's infected or not until a couple of years, until it's already, showing some level of blotchy, mottle, or chlorosis symptoms on the leaves. Of course, they could do PCR-based testing, but you can't do that in hundreds of acres of land, right? Yeah, early detection is a big challenge because of its poor growth, and these seasonal fluctuations exist in summer periods. I don't call it, it moves down more into the roots, but it clears more in the above ground, but it just, doesn't accumulate as much. And so in the summer, you see these low titers in the above ground tissues. So if you are only looking at above ground tissues, you may not pick up the bacteria. And so you need to sample different, I guess, leaf tissues, but also root tissues sometimes to see if the bacteria is in the tree.
Right. Right. Well, I think a couple of things come to mind. One is that just those challenges we talked about in detecting asymptomatic Africanus and, the low detection levels present some real challenges and barriers for importation, exportation, problems that aphids have to deal with, but certainly problems that, even interstate movement and thinking about those things are probably also challenging. As someone who studies, both insect and pathogens in forest systems, I know that we could use monitoring of the vectors to look for incidents on the landscape. But do we worry about new vectors emerging, or is it too co-evolved with its host for us to have those concerns?
The concern is more on, the insects. The main way they control the disease in the fields right now is through insect vector control.
Right.
We spray pesticides. We learned a lot from Florida. And so we are implementing coordinated sprays throughout the valley here. All our citrus growers follow a timeline, and that is recommended by our entomologists here in the valley. Now, there is a concern that there may be sometimes resistance to the buildup in the insects itself, and that could result in your insecticides not working, and there's not that many insecticide chemistries that we can use. So that's a major concern, then more so than other completely new vector transmitting the disease. There is related pathogens like Candidatus libribacter solanacearum that it is transmitted by a different psyllid vector. It's also a psyllid, but it's a different genus and species, Bactericeri coccarelli. Now, in that, we already have found that there are some resistant insecticide-resistant colonies already. So in that case, it's not a big problem yet in Texas. It has its cycles up and down. I'm talking about zebra chip disease now, because of the, sometimes the potato is a seasonal crop. Sometimes the psyllids are there, high levels, and sometimes it's not. So it's a big worry, insecticide resistance, more so than the bacteria being picked up by a different vector itself.
How do you think your work on zebra chip has helped your understanding of HLB and vice versa? Do you think working on HLB has helped your grasp of understanding combating problems in zebra chip? Is there synergy there?
Yeah, actually, good point. There is some, some commonalities between the two. As we just mentioned, they're both insect vectored and they're both phloem limited pathogens. So for me, studying zebra chip was really good, as in it's here, we have to study and the point of controlling it and finding ways to control the disease and the insect vector, but also it's a good model system. It's much more closer than obviously any other surrogates that are non-pathogenic, Cynorhizobium, or even the Libribacter crisna. This is zebra chip one is a pathogenic bacteria. So
Could you clarify real quick? So zebra chip is a problem in Texas, or is it a U.S. problem that you happen to be working on in Texas? What's the situation?
So it is a problem in US. In Texas, we have it. In fact, it is also up in the Pacific Northwest regions. May not be as much high, impact, negative impact as of today. I consider it as citrus greening from some 15 years ago. That's where zebra chip is today. Globally, we know that temperatures are rising. If potato psyllids start, moving to these newer regions where temperatures are warming up, that can quickly, transmit the bacterium to those new regions. For the most part, it's the weather that keeps it under check, the potato psyllid. So it is a problem in Texas. There were a few years ago, the populations were very high and our growers lost quite a bit. But then it has been down in the last 3 years since the weather has been, favorable for the growers,
I don't want to pretend that citrus and solanaceous crops are the only systems you work in. I've seen that you've done some work on spinach diseases, including anthracnose and white rust, which is not a rust, nor is it a fungus. Albugo is an oomycete. You've worked on sugarcane. Any other crops or systems that you're currently working on that are important to South Texas in your region?
Yeah, so some of the crops you mentioned are more in collaborations with our colleagues here. We have a interdisciplinary team. We have breeders, plant breeders, physiologists. So we kind of work as a team, and so our plant breeders who are working on the spinach diseases through, ways to introduce resistant traits into their cultivars. So in addition to those, you mentioned that there is also tomatoes. We work on tomatoes. A lot of projects in tomatoes in relation to not only breeding but also gene editing. We are using some gene editing to improve the vitamin C levels in collaborations with our breeders. I can think of vegetable crops, and we used to have a lot of sugarcane research. It has come down lately because of water problem. We are not having enough water in the valley to grow our sugarcane, so we even our sugar mill closed this past year, so it has been a little low, but sugarcane is an important crop for the valley here. And comes along with it is the energy cane. I'm not sure if you are familiar with the energy cane. It's
So it was just like the way we use like corn for ethanol. It's like sugar cane for a biofuel. Is that what you mean?
exactly like a biomass feedstock. In this case, it's a high lignocellulosic, sugarcane varieties have bred for energy purposes. And so there is a lot of research in the center related to energy cane as well. We have a breeder dedicated for that sugarcane and energy cane. So we work hand in hand with my colleagues and, extend my knowledge in genetics, genomics to all these other crops and, helping, either improving their resiliency to abiotic stresses, but also plant diseases.
I'd say your work on sugarcane is sweet, but some of our listeners might roll their eyes with that pun. So I want to just pivot back to HLB. Some of your work talks about developing therapies for HLB. What does that mean, and how would you deliver them to infected plants? I mean, I can grasp how you're using your hairy root system, but at the end of the day, how are you mass producing them for trials to test these things?
Great question. So, in fact, it's part of a 4-year coordinated ag project that we're testing exactly what you're asking. Let's say we have a peptide or a small molecule. How do you take it to the next step and apply it to these acres of fields, more in sense, at the practical level? For peptides, I'll tell you in two parts. First part is the peptide therapy. So the peptides is a gene product, as you know. It's a small 30 to 50 amino acid peptide. We can synthesize it, but the problem is costs. It's because it's so expensive to synthesize proteins and products. So we decided to put it in a genetic vector, but in this case, we used a viral vector, a citrus-specific virus, Citrus tristeza virus. Okay.
Okay, yeah, I've heard of that.
Okay, so this is a viral vector that has been developed by University of Florida, my collaborators there, the inventors, and they developed this system, Dr. Bill Dawson and his staff that are currently pursuing, they all pioneered that vector for citrus, we call it transiently expressing proteins in citrus. So we took advantage of that and we inserted our peptide or a gene, in this case, the gene construct corresponding to that peptide into this viral vector. And the beauty of the CTV or the viral vector is that you could easily, once you generate that construct and infect plants, you could take a mother plant and start taking pieces of that plant and graft it onto field trees that go into the field. So you can mass inoculate essentially your therapy with the viral vector mediated expression of the peptide into hundreds and thousands of trees.
And that's through grafting scion wood onto the field trees.
Exactly. And you could take a bark flap and, just, tie. Right now we're testing it is in the, budwood that go into the field. So you have young trees, but you could also do in the field trees itself. We haven't gotten to testing in the old trees, but we are testing with already pre-grafted materials that are going into the field. So basically the peptide is already expressing in the tree and almost like a transgenic. You start with the baby's transgenic plant and adapt it in the greenhouse and take it to the fields, right?
Right, right.
We have a industry support for this project. It's Southern Garden Citrus. It's a juice company, orange and citrus, but also its parent has companies US Sugar. So in Florida, they have been supporting this research. And recently, we also started teaming up with another company called Silvec Biologics. So they are based off in Maryland, and they are also picking up on interest in some of our technologies. So together, what basically we are doing is using CTV as a viral vector, delivering the therapy to field trees, and that's the product. the product itself is a combination of the viral vector and the therapy as a combination. So this is the first for a EPA. It's going through the EPA approval process right now. It will be the first one if it's get approved in for ag that there is a product like this. We know we use it viral vectors for research, but never it has been, commercially used as a plant and pest, disease control product. So that's the way we do the peptide testings, okay, or delivery into the field. If I can go into the second therapy, the chemistries, which is the small molecules we have found that has bactericidal activity against this bacteria, to deliver them into the tissues where the bacteria is living in the tree, which is phloem, is a challenge. In fact, it's really hard. You cannot apply by foliar application. It doesn't get into the leaves as easily because of the waxy cuticle of the citrus leaves. And so right now, growers and researchers have tested ways to basically inject trees by drilling into the trunk, a little hole, and then put a syringe in it, fill with your therapy, and essentially like an injection, right? We call it trunk injections.
Right.
It gets into the xylem, and somewhere at the exchange points to phloem, it moves into the phloem, the chemical. And there is already good evidence that the bacteria, can respond to that chemistry in the field trees through this injection approach, especially with tetracycline-based bactericides. So we're using that as our approach for delivering these new therapies.
Yeah. Okay. Thanks. That was great. And I know it's complicated, and it sounds like, you described it yourself as a high risk, high reward scenario. But I mean, we have to take risks to protect some of our crops that face immediate threats from climate change, invasive pests, invasive pathogens. So it's good that there's people like yourself working on it. I want to shift real quick. You're the 2024 recipient of APS's Syngenta Award. Do you want to tell me a little bit more about your relationship with APS and how they've shaped your career? Certainly, they produce this podcast, but I think a lot of us that are plant pathologists are where we are partly because of the support of APS.
Absolutely. I think, being part of APS has been great for me. Since my postdoc years, when I started working with plant pathogens, I started engaging with APS Society. As a postdoc, I was attending a lot of conferences. We attended workshops, and all the career, related workshops at the time were really helpful for me. But continuing as a faculty, I encourage all my staff and faculty to be actively involved in APS things. Tell you the truth, I'm not as active as I would like to be, but I do, I've helped this in phytopathology. I was associate editor and for since few years, and I do actively work with the Caribbean division. I organized last 2023 the Caribbean division meeting here in the South Padre Island here. We were really happy to see a good attendance here. So, I mean, being engaged with the society pays off in many ways beyond, just networking, but, it's about also connecting with your colleagues and identifying those research topics of interest. For students, for sure, it's a place to find their next jobs and next labs.
Certainly. I think it's good to point out that someone who's in a location like you are in extreme South Texas, it may be geographically isolating to attend some of the national meetings because they always, APS had its meeting in Austin, I think in 2012. It's not that Texas isn't a location
Yeah.
for the meeting. It's just that, if you're in Texas or Maine or Alaska, some of these places, it's hard to get to the national meetings without a lot of time and money. But involvement in the chapters, the branch chapters, is really important. I'm in the Potomac division, and some of the most rewarding things that I've given back to APS has been my involvement in that division. And it sounds like the Caribbean division is an important way to connect to APS for you as well.
Yeah, absolutely. I was there for several of Caribbean division meetings, and it's small, but we want to make sure that all the members, at least, work out their, networking and opportunities for the grad students. Yeah, I was there actually at the 2012 Austin meeting when I was still a postdoc. That was a good.
Oh, okay. Yeah. I organized the forest pathology field trip during that time, even though I was living in Virginia. But yeah, small world.
Cool.
I can't help but think about Xylella fastidiosa talking to you. It was believed for 100 years, 70, 80 years that it too was unculturable. And then they figured out how to culture it, if I'm not mistaken. So something that was considered to be really picky and finicky. They figured out how to grow it and grow it in pure culture. How close do you think we are to figuring out how to culture the causal agent of HLB in the absence of the hairy root system? Do you think we're within the next 5 years of figuring that out, or do you think it's a much longer time window?
It's really hard to tell predict the future, but there are a couple of active research groups that are really trying hard to get it into axonic cultures. And if one of them, I may have some good progress. I don't think they have published yet. And one group has already published one in biofilms. This is David Gang's group in Washington State. Several years ago, there was a publication. It was as a co-culture, again, as a biofilms, not a pure axonic culture.
Yeah.
But maybe if I would say from the industry point of view, citrus industry wants more to find the solution for the greening more so than whether you can culture it or not. But from a pathologist, scientist point of view, yes, that's really a roadblock right now. Maybe 5 to 10 years would be a guess for me.
Well, I see learning how to culture something, if it can be cultured alone, as a stepping stone for providing faster turnaround for solutions for solving this problem. We're about out of time here, but I've really enjoyed our talk, Kranthi, and I hope you have too. Thank you so much for joining.
Yeah, absolutely. Thank you.
We've just heard from Dr. Kranthi Mandadi, professor of plant pathology and microbiology at Texas A&M University at the AgriLife Research Center in Weslaco, Texas. As always, be sure to check out the show notes section immediately following this episode description for links to Dr. Mandadi's research. I'm Matt Kasson, host of Plantopia. Thanks for listening.